Okay, I think we can get this started. So I hope you can all hear us properly. If there is some problem with the sound, please let us know. And my colleague Adriana will let me know, and we'll try to sort it out. So welcome again to this webinar hosted by A4Cell Nano Devices. So my name is Julio Martin, and I'm Senior Strategic Liaison in A4Cell. And the title of the webinar today is Harnessing Innovation ROS, Reactive Oxygen Species Detection in Live Cells with the CytoCHECK SPAchip OHrad ROS. And we are going to stress an application on predicting cardiotoxicity induced by antitumoral anthracyclines and maybe other cases beyond. In A4Cell, we had a dream. Imagine that you could keep an eye inside a living single cell and be able to monitor and to use this device as a reporter, to know everything what is happening at every moment in time within individual single cells. So that was the dream. And here you can see these green squares. So let me introduce, these are the SPAchips to you in this particular image. So how are we doing that? So our approach has been to evolve the probes in solution, fluorescent probes in solution, to detect, analyze physiological hallmarks in the cell. And we think that by tethering these fluorescent probes to chips, to silicon chips, we will be able to overcome the drawbacks of dyes in solutions. So let me illustrate some of these drawbacks and how we propose to overcome these issues. So the first one is the cytotoxicity of probes in solution over time, which is due to the free access of these molecules to organelles. So these molecules are diffused throughout the cell, can get into organelles and make damage. So that results in a limitation in the experimental time, so that the cells can bear with the probe in. So by having the probe in solution, you end up with a reagent which is harmless and non-cytotoxic. Why? Because it resides only in the cytosol and covalently attached to the chip, so it's not a spread. So that will allow you to monitor in real time the cell culture, individual cells for weeks. The second limitation or caveat is the limitation of multiplexing of the fluorescent probes in solution, which is based on a spectroscopic resolution, whereas with the probes on a chip, so you can multiplex not only based on a spectral resolution but also in a spatial resolution. Here you have a cartoon of the chip with 4 probes grafted on top of the surface. So that's what I mean by spatial resolution. You will be able to identify where's the position of each probe, even if there is overlap in the spectra. So next property that we think we can overcome is the fact that these probes in solution can be metabolized and ejected out of the cell, by different mechanisms, cellular mechanisms. However, the probe on chip are not expelled. We have no evidence of these chips, once they are internalized, can be expelled out by the cells, and they remain metabolically stable because they are not accessible to organelles that can break this down. Poor cell penetration of probes in solution refers to the fact that each probe is different. So the penetration, the uptake is based on the physical chemical properties of the probe, and varies from probe to probe. So it's something that you have to optimize and interrogate in an ad hoc basis. So, however, the probes on chip are all internalized through the same mechanism. So we firmly believe that this is based on endocytosis, and very high chances are that this is in particular micropinocytosis. So, and this is irrespective of the probe, of the molecular probe itself. So this mechanism is served by all the probes. So that means that we have a universal way of getting the probes inside the cells. So this is our technology. Our IP in A4 cell is based on patents around this methodology and the fabrication and manufacturing of these chips. So we manufacture the chips as a suspended planar array. Actually, this is the acronym of SPA stands for. So suspended planar array is what you can see in this cartoon. So it's on a silicon oxide wafer. So you have the chips suspended, anchored to the wafer by a base. And then you print the probes on top of the surface. On the left-hand side of the slide, you can have a bright field image. So the chips are 3 microns by 3 microns wide, long, and 1 micron high. And I was saying that they are internalized by endocytosis once they are inside the cell. So the membrane, the chips escape from the endolysosome, and they remain freely accessible to analyze in the cytosol, as I was saying before. So at the bottom of the slides, you have a cartoon to illustrate how we manufacture the chips. So the chips are chemically functionalized, by different chemistries that we have, in order to afterwards have the probe that is going to detect the analyte attached covalently to the surface of the chip. Once we do that, we embed all the suspended array on a film, which is biocompatible, and then what we physically do is we peel off the film out from the wafer. So, and this is our reagent. So we dispatch is the biofilm that you can see here in blue inside an Eppendorf tube. So this is our reagent, and that's what you can see here on the right-hand side figure. So that's the solution of SPAchips. That's the first step. So you dissolve, you resuspend the chips on a buffer, which depolymerize this biofilm, and then you wash out and you resuspend in the cell culture media. Alongside this process, you have already prepared your cell plate with your culture of cells. So the first experimental step in your procedure is the pre-incubation of the SPAchip solution with the cell plate, with your cell culture. You incubate overnight. That's our recommendation. This is a process that could be optimized because it might vary from the conditions. The incubation time might vary from cell type to cell type, and as well as the ratio of chips, number of chips versus number of cells, in order to guarantee a reasonable percentage of cells incorporating the cells, at least one chip. So once you have these the cells already pre-labeled with the chips, so you treat. I mean, you subject the cells to your insult, to your challenge, to your biology, and that's what we call here in this cartoon the treatment. You incubate. Again, it will be dependent on your experimental design, and you read and you analyze. So because the chips are not on the molecular probe on the chips are not destroyed, You can repeat this as many times as you wish, depending on the photo bleaching of the probe itself, which we have seen that it can last at least 4 or 5 weeks, and of course the phototoxicity of your cell culture. So that's regardless, independent on the chip, SPAchip technology. So that means that you can run real-time kinetics non-invasively, and you can monitor what is happening in one single cell for many time points and over a long period of time. Here on the left-hand side, you can see this time lapse, this video. So let me draw your attention on this particular chip and cell, which is at the left bottom part of the figure. And you can see here in the graph, each data point is one time of the measuring the fluorescent intensity coming out from this particular chip. This SPAchip is one of the new developments that we are in beta testing, which is the oxygen detection of molecular oxygen. So here you can see that you can monitor what is happening inside this particular cell over a long period of time without destroying the sample. Okay, so that means that this technology is amenable to many different readouts, in sequence, so you don't have to keep parallel cultures data points. So that results in saving precious reagents as your cells could be. Just to give you a flavor on the applications. So this is just one of the ones that that we have developed with and tested with collaborators with the chips that we have already commercially available, which are pH and calcium. So in this particular case, we have spheroids in which the cells are labeled with the SPAchip for pH in green. So we can work with 3D biology. In this case, is calcium signaling. Here you can see intracellular mobilization by ATP as agonist of purinergic receptors. In this particular case is pH. You can measure intracellular and extracellular pH over the same sample, just because there are chips remaining in the extracellular media. So you can, by analyzing the same field in your microscope, you can monitor both intracellular or extracellular readouts, in this case pH. So the red, we have two different modalities. One chip is in green and the other one is in red. The one in red allows ratiometric measures, so it's more accurate and precise. And last but not least, is one of our latest developments, which is this multiplexed SPAchip for calcium and pH on the same chip. So on the left-hand side, you have the signal in green for calcium, and in the panel B, you have the signal coming from the SPAchip for pH in red. So it's the same SPAchip telling you two different readouts. So with after this introduction to the technology, to the SPAchip technology, so I will spend the rest of the webinar, letting you know about the new development that we have launched 1 month ago, several weeks ago, for the detection of reactive oxygen species. So the name of the product is OHrad ROS, and is based on the SPAchip technology. It's a novel way of detecting ROS because it's based in this groundbreaking technology as SPAchip is. And in particular for ROS, is selective, robust, and stable. And let me tell you what I mean by that. So it's stable because the chip is only active, and the probe on the chip is only active once has been processed by intracellular esterases in the cytosol. So that means that all the handling that you need experimentally prior to the internalization in the cell is stable, and the chip is not destroyed, and it can bear this manipulation. The chip, the SPAchip, is selective. So talking about reactive oxygen species is talking in a in a universe of different species, chemical species. So in this particular case, our chip is selective of the hydroxyl radical. So I will touch on this particular feature later on. The kit is insensitive to pH, so that means that it's robust to interference by other mechanisms different to activation of ROS, induction of ROS. So that means that signals from the chip are bona fide coming from induction of ROS and changes in ROS. So are not other pathways or cellular mechanisms that can be induced and affects to pH. So there are many, as you well know, probes in solution that are sensitive to pH. So that means that's what we mean by robust. And this is just an example, I mean, a cartoon illustrating what I was saying at the beginning of the webinar, which is the SPAchip technology. In particular for our ROS kit, OHrad ROS kit. So we have this step in which upon internalization, the fluorophore is protected. The future fluorophore, because at this stage is dim, so and you don't see fluorescence. So it is protected and oxidized inside the cell to produce the fluorescence, to trigger the fluorescence. Other than that, the rest of the technology is similar to other SPAchips. We have been inspired by this consensus statement, that was a couple of years ago published. So in the sense that talking about ROS is talking about anything unless you say in particular what you are dealing with, right? So any probe, any detection kit should ascertain which is the actual chemical species that is involved in the signal. And that's what we have done, right? We have tried to characterize our chip in this regard. In this figure, we illustrate the selectivity and robustness of the SPAchip. On the left-hand side panel, you can see the response of the chip to 3 reactive oxygen species. In this case, is the hydroxyl radical, hydrogen peroxide, superoxide, and ion radical at 2 different concentrations. And as you can see, only the hydroxyl radical is able to produce an increase in fluorescence on the chip. And this increase in fluorescence, as you can see on the right-hand side panel, is insensitive to pH over a range of physiological pHs, as I was saying at the beginning. So we wonder how fast this reaction between the redox reaction between the radical, the hydroxyl radical, and the probe on the chip is. We like to be as fast as possible. And in our hand, we can see that is almost instantaneous. We cannot measure in comparison with the slow rate limiting step. In this experiment, what we did is we generate at a slow rate the hydroxyl radical through the reaction between iron 2+ and hydrogen peroxide. So this is the slow rate limiting step, and this is coupled, to the. This experiment is in the absence of cells, right? So, and here is this radical is reacting, is going to react with the reduced form of the fluorophore, as illustrated in my slide, my former slide, and this is reacting. If this is very fast, I mean, the controlling at kinetic level, the controlling reaction is this first one. And that's what we have seen kinetically, right? So when we monitor the kinetics, we see that this is first order kinetic reaction depending on the concentration of hydroxyl peroxide. Here we have tested 5 and 1 millimolar. So the take-home message here is, as soon as the OH radical is formed, it will react with the fluorophore reduced on the probe at any place where it's present in the cell. It's true that a picture is worth 1,000 words. So here now is a series of slides with pictures of the SPAchip in action. So in this first one, so you can see an induction by this molecule, which is a quinone molecule, and you can see the SPAchips, these squares in green inside the cell upon induction of ROS in this fibroblast cell line. Here is a comparison of the extracellular, which are off state of the probe, versus the intracellular in the on state of the probe. So in this bright field image, so let me draw your attention on, for instance, these particular chips. There are other extracellular chips that you can identify in the image, but here you can see that in the fluorescent image you cannot see this chip detected, visible, whereas this chip, which is intracellular located, so you can see that it can emit fluorescent, upon excitation. So this is just to illustrate with an image what I was saying about the intracellular activation of the chips once it's internalized. This is a comparison with a probe in solution, and where you can see that unlike the previous images, here you can see that the fluorescence is much blurrier, and this complicates the image analysis. So the segmentation in the former image is easier. To give you a flavor of, again, the potential to interrogate 3D biology. So this is an example in which we have cells forming spheroids with the ROS SPAchip inside. So this is our preliminary experiments, but this is just to give you a flavor that, yes, indeed, the potential is there to work with 3D biology. Okay, so let me go back now to the induction by different chemical agents, and once of them being anthracyclines. One of the power of the SPAchips is that it's not only a question of qualitatively allowing you to measure what is happening inside the cell, is that you can precisely quantify, so was the extent of this induction, right? And that's very important. So what we have done in this experiment is we have challenged the cells with different agents and see whether or not it's dose response dependent, and it's time dependent. So here you can see doxorubicin at different concentrations. It's an antitumoral agent, as you may know. Here you can see a very nice dose response, a very well controlled standard error of the mean. And here you can see the panel at 6 hours. And in this particular case, at this concentration, you can see an increase. Although it's interesting that the other ones are not increased, maybe saturating the chip. Maybe I didn't say explicitly, but the chips and the probe on the chips is irreversibly reacting with the ROS. So that means that it can be saturated, once all the probe is reactive. And here you have other agents on the panel of the bottom. You have the 4-ball ester PMA, and you have a peroxide TBHP, and you have this quinone as well, which resembles similarity with the doxorubicin that I will illustrate later. Okay, so yes, you can see a dose response, you can quantify, you can see a time dependency, and there are more than one type of agents. So what they all should share at some moment is a molecular mechanism of generation of hydroxyl radical, and that's something that I would like to touch on in more detail in the following slides. We have compared with probe in solution. This is a standard, a traditional probe for ROS studies, H2DCFDA. In this case, we have compared with TBHP, and you can see that the signal to background is higher for the chips than for the probe in solution, which is also advantageous. So why? Yeah, let me discuss with you the molecular mechanism of detection and the cellular mechanism of generation of ROS, and why these agents can be detected. That's our hypothesis. And here I told you in the formal slide, in the formal figures, that PMA, the 4-ball ester, doxorubicin, and a chemically similar similar compound, which is a quinone, are detected by the chip. So that means that it will generate hydroxyl radical. How? So it's described that PMA is able to activate NOx, NDPH oxidases, to produce superoxides. In the cell, you have superoxide dismutase that generate hydrogen peroxide. And hydrogen peroxide can react with iron to give hydroxyl radical. This is the well-known reaction, Fenton reaction is called, right? So this is a mechanism that is linking NOx, PMA through NOx to OH radical. In terms of anthracycline, here you can see in small, I'm going to zoom this in. So what is called anthracycline redox cycling. So that means that there is a mechanism in which oxygen, superoxide anion radical can be formed naturally, spontaneously. So, and this will follow the same route or pathway that I was describing before. So this can be produced in the cytosol and also in the mitochondria. So this results in the formation of hydrogen peroxide, and then through Fenton reaction, all the mechanisms converge to this generation of hydrogen peroxide and the conversion to the radical through the Fenton reaction. So here you have a zoom for the particular case of doxorubicin and analogs of the same family. This is a complex, slide, but just the take-home message is that doxorubicin, through this redox cycling and as I said, analogs, can produce superoxide, hydrogen peroxide, and then through the Fenton reaction, which is a common mechanism, produce the free hydroxyl radical. And there is a parallel mechanism in which the anthracycline in particular can generate OH radical without any enzyme being involved, so directly reacting with the hydrogen peroxide. So there are many mechanisms converging to the generation of OH. And here you can see the importance of or the impact, the relevance of OH generation in the health of the cell. OH radical is a very reactive species. I will give you some figure later. That can react with any molecule, any kind of molecule. Actually, it seems that it said that it can only travel in the cell a few angstroms, right? And it can generate lipid peroxidation, protein modification, DNA damage. So it's a quite orthodox reactive oxygenous species. And don't forget about lipid peroxidation, because this is something I would like to stress later on. So you have another ways of non-enzymatic ways of generating the free hydroxyl radical, which is the Fenton reaction coupled to the Haber-Weiss reaction. So it means that, yeah, by the superoxide anion radical plus hydrogen peroxide, you can convert this into the OH radical without the participation of any enzyme. So it's a quite divergent way of producing hydroxyl radical. And here I was telling you about peroxidation of lipids and phospholipids, and what's the implication of OH and the importance of OH in cell health and the link with diseases, right, and therapies. And here is just to give you a flavor on this and to illustrate this importance. So ferroptosis. So ferroptosis, the term was coined 2012, so it's a recent term, although something similar events had already been described. So it's a programmed cell death independent of apoptosis, the classical apoptosis, which is dependent on iron and ROS. And as I was telling you, iron, ROS, the Fenton reaction, hydrogen peroxide. So it's generating hydroxyl radical. And here you see in the middle of this cartoon, the pivotal role of iron and ROS in phospholipid peroxidation. Without getting in all the details here, it's important to stress the importance nowadays in therapies, in the disease, because it can cause destruction of and death of cells, has been linked to neurodegeneration, for instance, Alzheimer and Parkinson, for instance, cardiac diseases, but also in the induction of cancer. I mean, the induction of ferroptosis to be used as a therapeutic approach to cancer in tumor cells, right? So ferroptosis is important. It's a programmed cell death mediated by free hydroxyl radical, and our SPAchips could be of value to study this phenomenon. So I was illustrating in the former slides the key role of at the center of all the ROS mapping of hydrogen peroxide. Why is that so important? Maybe the most important ROS, and it's because of the stability. So hydrogen peroxide is the only stable ROS species. So if you compare with the OH radical, which is just nanoseconds, so that means that as soon as this form is going to react, only a few angstroms to travel in the cell. The superoxide anion radical is only a microseconds scale. However, the hydrogen peroxide can travel, within the cell, and it can also penetrate the cytosol from other organelles and can go across membranes, lipid membranes, by facilitated diffusion, right? So that means that hydrogen peroxide generated in another organelles, not in the cytosol like in the ER or in the mitochondrial, can go into the cytosol, and then through the Fenton reaction can generate the free radical. So you might be wondering, okay, I'm working on some particular cell biology phenomena or problem in which it's not the hydroxyl radical is not the primary species generated. Can I use your chips for my research? And the answer is yes. Chances are that you will be able So you have to make sure that at the end of the day, as a secondary mechanism or species downstream of the first primary generation of your ROS, the hydroxyl radical is generated and formed. So hydrogen peroxide can be seen as the Trojan horse, right, of ROS and production of OH radical, in other parts of the cell. And this is just as a summary. This cartoon is a quite simple summary. That means that it's going to work in my system, in all the systems? No. Why? Because there is a competition. There's a competition between different mechanisms of removal of hydrogen peroxide. So if the Fenton reaction is predominant over the other ones, high chances are that you will be able to detect it. If there are other mechanisms which are important, much more important kinetically than this one, the Fenton reaction, so maybe not, because they are not going to generate the free hydroxyl radical. Okay, so I would like to, I'm going to stop here. We have 20 minutes. The technical part of the ROS SPAchip detection kit with all the features and properties. But before opening the floor to the virtual floor to questions, to Q&A, yes, I would like to tell you a few words about the company, A4Cell. It was founded in a startup based in Spain, nearby Madrid. It was funded 6 years ago. And as I said at the beginning in my first slide, our dream is revolutionize the living single cell analysis with the lab in a cell devices, and ultimately our commitment to discover new drugs, to help the researchers and drug discoveries to develop new therapies, as well as cell biologists to ascertain the rationale, behind diseases. Our portfolio of products is pH, calcium, multiplex, and the brand new ROS. Next developments. So we have in beta testing, we have a probe for molecular oxygen, intracellular or extracellular, and we are working on different versions of multiplex chips and combinations of the single detection kits. And with that, I'm gonna stop. I will be more than pleased to to address your question. Please post them if you haven't done it yet in the Q&A, and I will go through then. Otherwise, drop me an email at any moment, and we'll reply you as soon as we can. So I'm going to stop sharing, and hopefully I will have access to the chat or the Q&A, and try to read if there is already some question there. I don't see questions in the chat, maybe. Yeah, here in the chat, there are some questions, I think. There's a question saying, how the biocompatibility of the chip has been analyzed. So the first was the rough cytotoxicity studies to see if there is a change in the cytotoxicity. No changes, no decrease of viability of cells over these 4 or 5 weeks incubation. The other readout we use is mitochondrial health. Again, is healthier than any other probe in solution that we have tested. And as we speak today, we are conducting studies on proteomics and transcriptomics. The preliminary output from the proteomics is there is no particular pattern of toxicity, on the cells. Which oxidation agent has been used during the internalization? I don't know what you mean by that. Which oxidation agent? I cannot tell you. I know if you want to know details about the chemistry itself, it's something that is IP sensitive, so that's something I cannot tell you. We have a patent file for this SPAchip. I don't know if you mean that. Otherwise, I cannot capture the meaning of the question. The next one is OHrad ROS SPAchip reversible? No, it's not. The detection is based on a irreversible redox reaction. So once the probe is oxidized by the ROS species, is not going to be able to be recycled. For how long can you measure? What is true that if the signal once it's formed is stable, and we have monitored this for days. No, in this particular case we have not extended the experiment for many weeks, but for days, as far as I remember, maybe 3 or 4 days. So you can see the signal on the chip stable. And the extent of the experiment, as I said, it will depend more on your cell culture, your cell type. Doxorubicin is highly toxic to the cells. Do you normalize the fluorescence to the amount of alive cells? That's a very good point. So we haven't done it in the figures. That it's a very good question. That we haven't done in the results I have shown you. Is we haven't done that. So I show you is the intensity for individual chips as an average, but they are not normalized. So bear in mind that the power of this technology is that it will allow you single cell detection. So it's not like in other maybe probes in solution that you can use macroscopically, in which, I mean, it's non-brainer. Something is a must. You must normalize by the number of cells. In this particular cells, you are detecting for a cell which is viable and live. So that means that this response is genuine and is meaningful. I don't know if there are more questions. If you want to make a questions, now, I mean, please do it. We can unmute you, I think, if you want. And if no more question, maybe we can close the webinar. As I said, I mean, please don't hesitate to share with us your, comments, doubts, questions, suggestions. We'll be more than pleased to receive your feedback. There's another question here. What's the relevance of being pH insensitive? That's what I meant. It will depend on the probe, the alternative probe for comparison that you are using. I mean, if there are probes that are the fluorescence is pH sensitive, so that means that this is the probe that you are using for ROS, and your challenge, your insult in the cells is producing a pH change rather than a ROS, or in addition to a ROS. So you are mixing signals. So it might be a misleading readout, and conclusion interpretation. So if this probe is pH insensitive, that means that all the change in fluorescence will be due to a ROS change. So it's more reliable and meaningful result. Okay, thank you very much for your attention. Hopefully you have found this interesting. And if you want to know more, so as I said, drop us in email. Thank you very much.